kicking things. So this is the first intensive period of the workshop. Glad to be here with Diana to get you started. So we're going to be talking about the 16S analysis pipeline. So just a reminder that the materials here are licensed under the Creative Commons Attribution Share Alike. So feel free to reuse, just let everybody know where these came from. All right, so here's what we're going to try and achieve over the next 2 hours, followed by lunch, followed by 1 more hour. You should be able to understand the main strengths and weaknesses of 16S, read and interpret the contents of sequence files, everybody's favorite, describe the process of sequence clustering, which you'll get from Diana, interpret the results of analyses, including boom, 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 taxonomic summaries, diversity analysis, and differential abundance. And for the tutorial session, you'll be able to basically do all these things. So conduct an end-to-end microbiome analysis using QIIME 2, and then know the main tools available to conduct statistical and diversity analysis. So just and here's the plan. So we've got a few different sections. The first one is just an overview of 16S and taxonomy. Then we'll talk a little bit about sequence data and quality control. Diana's going to talk about how to cluster your sequences. I'll tell you a little bit about diversity analysis, and then we will unleash you on the tutorial, which you'll be able to work at your own pace. Diana will be there to answer questions, and at any point, if you have questions, I'm happy to answer them in Slack or try to answer them. Then you've got the 1 hour lunch, and then in theory, Diana and I are going to split the hour after lunch between a theoretical bit on statistics and machine learning, and then some more hands-on practical. We have a lot of material and we may not make it that far, but that's fine. And I mean, the statistics and machine learning stuff at the end goes beyond the practical anyway. So if we have to shave some of that off, it's not a huge deal. And you've got the slides if you want to refer to them. And again, ask any questions. Quick note, how many of you have used QIIME 2? Look at the room. Show of hands.
There's 3 hands up.
Okay. I see a couple of hands. Great. Awesome. So the QIIME 2 software suite is a series of analytical tools for microbiome analysis, particularly with 16S. And so it can do everything from load in your raw sequences, do quality control, demultiplex your sequences, diversity analysis, taxonomic classification. It hooks into things like muscle and Raxameller fast tree for IQ tree for phylogenetic analysis. Has all sorts of nice visualizations as well, statistics, machine learning. So that's all great, and it's a very useful resource, but it's important to keep in mind that it's not the only game in town. And so particularly for some of the more exotic, potentially interesting analyses that you might want to do, you may find yourself going into, let's say, our libraries or our packages or other standalone applications. So we're focused on QIIME 2, but just be aware that it's, as I said, one of several alternatives. Okay, so why 16S? So you're all familiar with taxonomy. Basic idea, classification of organisms according to a predefined hierarchy. And we all know about kingdom, phylum, class, order, family, genus, species. But what's interesting, well, one of many things that's interesting, is that the history of microbial taxonomy is big and confusing and messy. And at various points, people have believed either that there is no taxonomy because many bacteria are just the same bacterium showing us different faces at different times, or, and you see this in the 60s, people just throwing their hands up in despair and saying, I give up. We're just going to name stuff, but any notion of relatedness among taxa, forget it. Brief overview of different criteria people have used to define taxonomy. Marker gene similarity is underlined because for a while, your typical species definition rested on the similarity of their constituent 16S sequences. So prokaryotic taxonomy has an interesting and fun history, just including one of my favorite examples here, 1988, the Proteobacteria. And what's cool is that the Proteobacteria were named for Proteus, a god of the sea that could take on many forms, because the characteristic of the Proteobacteria was that they could take on many forms. Thanks. But these days we find ourselves using genomic sequences and genomic sequence clustering and things like average nucleotide identity to cluster our genomes and also to infer phylogeny and do taxonomy on top of that. And so this is great. This is fantastic. One useful piece of this to consider is that there are lots of objective characters to compare, right? Nucleotides, amino acids, rather than having to make really crucial value judgments on, is shape a key defining characteristic? Is metabolism a key defining characteristic, etc.? Now you've got a bunch of characters that kind of all look the same. You can use models to describe their similarity and their evolution. More phylogenetically informative than morphology, can be assessed in an automated way. Let's sequence a bunch of stuff and either work directly with nucleotides or, in case of protein coding genes, infer protein sequences and work with those. So nice. And this, cell shape is a great example of this. Many traits that you might look at are actually subject to convergence, right? So not every spherical thing in the tree of bacterial and archaeal life shares a common ancestor that was spherical. So the convergent evolution piece is going to be less of a confounder. It's not going to not be a confounder. And if you've ever worked in phylogenetics of various weird and exotic things, you'll know that convergence can be a problem that can really mess up your phylogenies. So sequences are better for that, at least. However, genomic taxonomy is great with some limitations. However, in practical terms, if you're trying to characterize a microbial community, it's really hard to get the genome sequence of everything in order to perform this classification and this attribution in anything but the simplest of microbial communities. So, oh my God, I forget her name, Jill Banfield did some fantastic work with acid mine drainage communities in the 2000s that have like 6 things in them. So that's a bit easier. It's still not trivial. But for anything like a human microbiome sample or ocean water, forget it. So we need to choose a smaller sequence that everyone has with enough characters to distinguish different groups. And so Will touched on this briefly in terms of different sequencing technologies, right? So how many nucleotides can we profile in a really simple way that is consistent across organisms? It's important to keep in mind too that variation in sequence may not have much to do with function, right? And this is true as 16S, which I'll introduce in greater depth in a moment, cannot tell us about important evolutionary events. But in the end, what we really care about here is taxonomic assignment. So maybe that's okay. And the other thing is molecular evolution is weird. I'm not going to get into the details, but suffice it to say that it doesn't always behave the way you expect it to. All right, so let's say we want a short sequence that we can use to profile everything, right? All of the microbial biodiversity in our sample. Well, it stands to reason that perhaps the best way to do this is to actually identify something like a gene, some kind of stretch of nucleotide sequence that everyone has, more or less. And so people at various times have tried to profile the core set of genes in bacteria and archaea. And many different studies have done this, and it's just, different ways of defining the core, whatever. But just to cite one example, a method that was used for genome phylogeny called Amphora, it's made a while ago, identified just under 30 conserved core genes across all of microbial life. You see them there. The ones that are highlighted in red encode ribosomal proteins. So you can see that we've got 23 in red, and all of these encode proteins that are part of either the large or small subunit of the ribosome. Well, okay, isn't that interesting? Maybe we're starting to get somewhere. This is great, but there are issues with protein coding genes. One in particular is that of the genetic code, because your typical pattern in a protein coding gene will be highly conserved, not so conserved, highly conserved, not so conserved. And so fine, that's a typical sort of codon pattern. But what it means is that it's difficult to design primers that span the entirety of life, because every third position, roughly, will have some degeneracy. And so designing primers to amplify that gene specifically could be tricky. People often use specific protein coding genes for smaller taxonomic problems, but in general, not so much for the tree of life. So what else can we do? Well, we can take a closer look at the ribosome. The ribosome remains a great target. Everybody has it because everybody needs to make proteins. So it's a good thing to focus on. So the prokaryotic ribosome has approximately 45 proteins split between the large and the small subunits. I say approximately because it depends. There's all sorts of variations there, but roughly. But in addition to this, we have these 3 ribosomal RNA molecules. So there's 2 in the large subunit on the right, and then there's 1 in the small subunit on the left. And it turns out that for various reasons, the 16S ribosomal RNA gene is a good choice. Part of these are historical. I won't get into it, but it's got a good length and it's got certain useful patterns of variation that make it a good target for fairly small scale taxonomy, and then taxonomy that spans the entirety of microbial biodiversity. And so I've showed you a little map here of the 16S ribosomal RNA gene from nucleotide position 0 to nucleotide position 1500. Again, there's some variation among species, but the key point generally remains the same, that you've got different regions. And so you can see those blue regions and you see the red regions. These are differentiated because the blue ones tend to be much more highly conserved and the red ones tend to be much more variable. What this means is that if we're trying to profile a microbial community, and if we're trying to do it using something like PCR, then we can design primers against, generally, those conserved regions, because the more conserved the region, the more consistent it will be across huge swaths of biodiversity. So the more we'll be able to use a single or at least a small number of PCR primers to make the problem tractable. So that's great. We have these places where we can target primers. But the other nice thing is that the variable regions give us some amount of variation that we can use to get fine-grained taxonomic resolution. And so examples of this include primers that will amplify everything from variable region 1 to variable region 3, or V3 to V4, V3 to V5, and so on. V6 to V9 is a truncation artifact, but it should be a bit longer than that. And so the point here is that by using different combinations of primers, you can get different regions of the gene amplified. And people have their favorite primer pairs. Sometimes you'll see standards in different communities, meaning research communities for different types of habitats. But ultimately, you can use primers to get this taxonomically informative sequencing done. Okay, and if you're using a big fancy technique like nanopore or PacBio, you can actually sequence the whole thing, which obviously is the best in terms of finding a good number of informative characters. But as Will pointed out, the error rate for PacBio and nanopore tends to be a lot higher, which can really confound your taxonomic classification, unless you do some fancier things with the sequencing technology. It's important to keep in mind as well that although we typically represent it as a straight line, 16S ribosomal RNA is not in fact a straight line. And so there's a nice little map here from a structural study that was published a few years ago, where you can see the secondary structure of the ribosomal RNA molecule, where the complementary strands go and stick together, where we get these stem regions, which tend to be the ones that are more conserved, and these loop regions, which tend to be the more variable parts. And you can see in the colors there, the different mapped regions. So the story of 16S. It's good because everyone has it. It's good because it has these conserved and variable regions and none of this genetic code stuff that we need to deal with. As I said, part of the reason people use 16S is because people use 16S. The primers are well defined, things have been studied, different variable regions are reasonably well understood, and an absurd number of sequences can be found in the public databases. So I checked yesterday, and I think this is still the current number, over 9.4 million 16S ribosomal RNA sequences in the latest release of Silva. Fantastic. Limitations. It's less good because many genomes have multiple copies. So you look at many isolates, many strains of E. coli, and they can have like 6 copies of the 16S. So think for a moment about what that does to your quantitative estimates from a given habitat, right? E. coli K12MG1655 has 6 copies. This other organism might have 2. So you're going to get weird estimates because of that. At the finest levels, like strain level resolution, you will typically not get enough resolution. And you'll see this when you start doing taxonomic classification. Things really start to fall off when you start talking about species level. The other thing is, as I said, people have universal or nearly universal primers to amplify these things, but there are still sequence differences. And those sequence differences can lead to amplification bias, which means that certain taxa will be overrepresented in the sequence sets you generate. And so this is not a fault of 16S, but it really is an artifact of different primer affinities. Oops. There we go. Quickly, because I feel morally obligated to complain about taxonomy a bit. If you're trying to do taxonomic profiling, what's the rank you want to describe? People often talk about species, but species are in many cases not really a thing. And if I tell you, hey, you've got E. coli, well, that could be anything from deadly pathogen 0157H7 to happy commensal K12 to probiotic strain Nissel. So there are limitations in the descriptive power. You might want to summarize at higher levels for reasons like the statistical support I just explained, but then you lose a lot of information because then you're not saying, hey, you've got E. coli, you're saying, hey, you've got gamma proteobacteria, which is an even less meaningful thing to say. The context may define the interpretation or may influence it, right? So, Pseudomonas. Pseudomonas is a really nasty pathogen in diseases like cystic fibrosis. However, Pseudomonas can be found in all sorts of habitats, and so your interpretation of what it's doing may depend a lot on the habitat you're looking at. Sometimes, and you'll see this in the diversity section, we really care less about the number of things we identify, the number of species or phyla or whatever, than we do about the specific identity of the things. So that's, that's maybe a useful out in some cases. Just to finish the act. So this was a paper published last year. I don't know how many of you have heard of Pseudomonadota or the controversy around Pseudomonadota, but there was this paper last year that renamed 42 bacterial phyla, caused a bit of a firestorm in the community. And so this is just one example, and Campylobacterota. I love it. The properties of this taxon are described by replacement of the illegitimate name epsilon proteobacteria, which is an earlier synonym for Campylobacterota, but is illegitimate as it was based in the illegitimate class Campylobacteria, which is a later homotypic synonym of epsilon proteobacteria. You may not want to get too deeply into this, but just to say, here be dragons. All right, we're way behind time, but if anybody has a question, maybe we'll just take a pause and somebody could put their hand up and relay it, or just post something in the Slack. Oh, I see that somebody has posted something in the Slack. Oh, it's a thumbs up. Question?
No.
No. Okay.
No.
T'as l'air un peu déçu, là, Diana. Anyway, on continue. Okay, so the second part is really more about a means to an end, right? Sequence data, sequence retrieval, and quality control. So quality control is a thing we have to do because DNA sequencing is messy, right? It's error prone, you get truncation artifacts, you get systematic errors. Things can just go wrong for whatever reason. And so the first thing I'll mention is that there are lots of different places you can get 16S data and other types of data, of course, as well, public databases. I mentioned Silva already. There's a list of other ones. And then you often also find habitat-specific ones. So HOMDE, Human Oral Microbiome Database, is a good example of this. So lots of places to retrieve sequences. Interestingly, we have some that are sort of in principle redundant with one another because Silva Green Genes RDP, Ribosomal Database Project, all give you taxonomic information. And so the question is, well, which one should I use? I just include a tweet from Pat Schloss, who's the primary author of Mother, saying Green Genes is basically dead, which I think is still true. It was last updated, excuse me, almost 10 years ago. RDP is based on the classic Burgess manual. Could flip a coin on RDP versus Silva for classification. Okay, that's actually not what we found in our experience because Silva's a lot larger. We tend to get better representation of a lot of taxa of interest. We've seen this particularly with the vaginal microbiome, where some of the lactobacillus we see with RDP don't really make a whole lot of sense. So could flip a coin, not so much. And I think this is doubly true as people move to or not move to or complain about this recent renaming of 42 bacterial phyla. One cool resource that I wanted to point out is this Rescript, which allows you to do customized database retrieval and management. If you want to have nice reference databases that are maybe a bit more tailored, strongly encourage you to check it out. Okay, so how many of you have seen a FASTQ file before? Okay, I see more than 3 hands. So then those of you who have had the pleasure will know that there are 4 different parts of it. And for those of you who haven't, there are 4 different parts of it. And it's a repeating series of records. Each record has an associated header line, then the actual DNA sequence, and then the plus character, and then a series of quality scores. And so here's what an example looks like. The header can be kind of whatever you want. Typically, it has information about the run and sample name and so on. The second line is the DNA sequence. You may recognize it as an alphabet, a string on an alphabet of 4 characters. There's that plus sign. And then the last line is a bit weirder for those of you who have never seen it. So it said Fred quality scores. Well, a Fred quality score is basically a measure from the sequencing device that expresses how likely that nucleotide is to be wrong. So higher Fred scores are better, and the actual quantitative scores are reflected in each of these characters. Fred scores are numeric, but in order to preserve the one-to-one mapping from like that first A has a quality score, the first C has a quality score, the G has a quality score, each Fred score is represented as a single character. The quantities are based on the ASCII system, which is the numeric codes for each of the characters that you can type out on your keyboard. And so, for example, if we look at that first C, the ASCII code for C is 67. Now, in the Illumina 1.8 and up scoring description scheme in FASTQ files, you would subtract 33 from that code to get the Fred score. So a C corresponds to a Fred score of 34. And so this gives you the basis for actually looking at profiling the quality of your sequences and making decisions about what deserves to stay in and what does not. So you need to import into QIIME 2. Not going to spend too much time on this because you're going to do it in the tutorial. But in general, your sequence data will be in FASTQ. If you have paired end data and you're presenting it as paired end data, then you will want to have each of the 2 associated files with the same name, just one having R1, the other having R2, so that QIIME knows that these 2 things are magically connected to each other. You can see these file extensions here, these fastq.gz. These are zip files, but you can still interact with them using commands like zmore. Okay, so this is fastq from a real file that we have. This is a vaginal survey of preterm infants in Kenya. And just to reiterate, this is an R1, and then the first 4 lines of the R2 correspond to the other direction. So the first record in the R1 file corresponds to the first record in the R2 file. So QIIME2 can say, oh, these are from the same 16S sequence, hopefully, almost certainly. All right, so as I said, your sequencer can lie to you. So you can ask yourself, is this a real sequence? Now, you generally wouldn't do this as part of analysis, but if you have skepticisms, then you can always take a given sequence and compare it against a reference database of 16S. So here I did that. I just blasted the sequence I showed you before against RefSeq, and I found a really close match. So the query line for BLAST is the sequence I just showed you. That's from our study. The subject is the closest matching reference sequence. And so in this case, we have a match. Oh, and I made a mistake, probably not the only one. Notice that in the BLAST result, it says Lactobacillus crispatus, which is a well-known vaginal associated microorganism. On the left, I said Lactobacillus plantarum. So it is actually crispatus. Those sequences are really similar, so it's pretty convincing that we have a 16S sequence. But notice there are 2 differences. I've highlighted them in red, where the query and the subject have different nucleotides. Are these real? Are these sequencing errors? We'll see a bit of that later on. Okay, so here's your commands for importing into QIIME2. Again, you will run this when you start Diana's tutorial. And QIIME wants all the fastq.gz files to be in the same directory. In this case, you can see the input path flag pointing to import_to_QIIME. It's useful to visualize your sequence quality. And so in this case, what you're looking at is an averaging over all of the reads in a given sequencing run. And so this is the first 40 nucleotides in this case. This is a fairly old figure. It's from the FastQ software, but I like it because it's got some nice subjective stuff in here that may be helpful and clear. And so on the left-hand side, you see these Fred quality scores. So in this case, from 0 to 34. At some level of Fred quality score, you'll say, I am done. I do not want to include these things. And if you look at this, you can really see how from the start of the read, position 1, to the end of the read, position 40, you get this steady decline in average accuracy and this huge increase in the variance of the accuracy. And so this really suggests that there are a lot of reads that have quite low accuracy scores towards the end of the read. And so depending on the filtering you set, and again, this will be in the tutorial, you may end up discarding many nucleotides from a lot of these reads. You'll probably end up discarding a lot of these reads as well. And if you see terrible quality across the board, well, you know you've had a bad sequencing run and probably should just stop there. Okay, and this is just a corresponding example that's generated from QIIME2's demux. And what's nice is that it's interactive, right? So you've got, you can look at this online or through QIIME's interface, and you can actually see your various statistics. Okay, so files are imported, you've done that. If the files were multiplexed, so if you had multiple runs in a sequence file or multiple samples in a file, then you can use tools like cutadapt to assign sequences to samples according to barcodes. You can also remove reads, again, that's sort of QC filtering. And then what's commonly done now is the data2 clustering algorithm will actually do some of that quality-based analysis as well. Okay, speaking of clustering, it is time to hand over to Diana. While Diana switches over, are there any questions about that? Hearing none. Okay. I will mute.
Sorry, Rob. I closed it. But if there are any questions while I switch my screens, you guys can type it in Slack or just raise your hand. No questions? Rob, can you hear that? She said, is there a difference in sequence quality between your R1 and R2?
Well, I mean, Diana, you've stared at this a lot. I think in general that the reverse read tends to have lower quality than the forward. And actually, I think your most recent results really do bear that out. I think those are on your poster too, right, for the conference?
Yep.
Okay, so yeah, generally the forward read has a higher quality than the reverse read. I see a question about explaining the quality visualization. I'll touch on that briefly. I don't know if it's still visible to people in the class, but if you've got the presentation in front of you, I'm looking at slide 24, which is the visualizing sequence quality. And so this will be a summary over many, many reads. And in this case, you can sort of treat it as if each of those reads is 40 nucleotides in length, right? So position 1 is the start of the read, position 40 is the end of the read. And so for that entire set of reads, we can look at each position. So we can look at the first position in read 1 and the first position in read 2 and the first position in read 3. Look at the quality scores of each of those, calculate the mean and the standard deviation. And so that's what you're seeing here. So for position 1 in all of the reads, the mean accuracy, the mean Fred score is 33, and you can see the variance of the standard deviation there. And so as we go from position 1 to position 2, you can see that there's a bit of a drop on average. This is not to say that there won't be some reads that are awesome from start to finish, but looking at this summary, you can see that many of them really drop off as we get towards the end of the sequence. You suggest R1 and R2 to overlap with each other when designing the primers to improve the quality. It's a good question. And I'm not sure what I would say is a recommendation, because there's a trade-off, right? If they overlap, then you get some nice checking in the middle. But if there's no overlap, then you get somewhat longer sequences potentially. So yeah, I can't really suggest something one way or the other. There may be others among the instructors who have stronger opinions about this, in which case I would defer to their expertise.
Thank you, Morgan. You'll see in the tutorial we're working with today, we have primers that have an overlapping region, but sometimes you'll have primers that don't have one, and you can just concatenate your reads, and that can work too. Are there any other questions?
think in the interest of time, maybe Diana, you can go on, and I'll keep an eye on the various chats and try to answer things. If anybody has any questions in the room, then I guess they can ask with actual speaking.
Sounds good. I'm just going to remove this. And I think, am I speaking loud enough, or would you prefer having me use this? Okay. Is it better now? I think it is better. All right, so I'll just do a short little part right now on sequence clustering. So this follows very well with what Rob was just talking about. Great. So the learning objectives of this short section is for you to understand how to cluster your marker gene DNA sequences and to be able to infer the taxonomic compositions of the microbial communities you're working with. I hope that by the end of this short section, you'll be able to tell me what's an OTU, what's an ASVs, and also to know all, or not all, but some of the available tools for your analysis and some of the limitations of each one of those tools. So this part is divided into 3 smaller sections. The first one, we'll talk about why we cluster our sequences. The second one, how we shifted from OTU, which was the traditional way to cluster our sequences, into ASVs. And the third part will be about taxonomy classification. So just a quick reminder, if you're interested in a microbial community, what you want to do is grab a sample of that microbial community. You want to extract the DNA from it, and then you amplify a region of interest. You can amplify a region of interest and then sequence that region. Once you get your sequences back, you want to cluster your sequences. And then once you have your little clusters of sequences, those will be the basis for your diversity analysis. So why do we cluster sequences? So there's a couple of reasons why you want to do that. The first one is to reduce the dimensionality of your data. So once you get your sequences back from your sequencer, and Rob showed what a FASTQ file looked like earlier, right? You have a lot of different sequences, and those files tend to be very, very large. So one important part of the clustering is to take all of those sequences and group them by what we would, or what the algorithm thinks belong to one microbe. So instead of having thousands and thousands of sequences, you end up with only some thousand groups of sequences that would each one represent one microbe in your community. So you go from very large file to a relatively smaller file. You also want to remove sequencing artifacts. So a bit earlier, Rob talked about sequence quality and how sometimes there might be errors that just slip into your sequences. So when you cluster your sequences, that's a good thing, because once you cluster 2 sequences that look a lot like each other, and one of them might have one error that slipped inside. So when you cluster them together, you might get rid of those errors and then avoid just having false interpretations of just sequencing errors. And also it can compensate for within species variability, because there are some species that have multiple copies of your gene of interest, and sometimes there might be variability within that same organism. You wouldn't want to consider that as 2 separate organisms, so clustering your sequences will allow you to just put them together. So let's talk about what's an operational taxonomic unit. So, or you can call them an OTU, you might have heard that word before. So an operational taxonomic unit is the word we traditionally use to call a cluster of DNA sequences. And just to show you how traditionally we used to do that, I tried to make a small little representation of that. So here we can see, oh, I don't know how to remove this bar. We'll put it there for now. So if we look at 2 sequences, let's suppose you got all of your sequences back and you want to look at forming clusters for your sequences. If you would use operational taxonomic units, what you do is you cluster your sequences based on a threshold of shared similarity between sequences. So if I look at this pair of sequences, for example, and assume that the total length of these 2 sequences is 100 nucleotide, and there's only 2 differences between these 2 sequences. To find the percentage of identity between these 2 sequences, we'll just do the total number of matches between the total length of the sequence times 100. And for these 2 hypothetical sequences, it gives us a percentage of identity of 98%. If for my analysis, I want to form OTUs and my threshold is 97%. It means that anything that shares 97% similarity or above will be clustered together, meaning that these two would not be clustered together. But if I'm interested, for example, of a more finer scale diversity, I could put my similarity threshold at 99%, and in that case, these two would not be clustered together because they only share 98% similarity. So let's look at what it looks like if we have this hypothetical microbial community composed of 4 bacteria, A, B, C, and D, that are 4 different colors. Let's suppose you sampled this community, you've extracted the DNA, amplified your region of interest, and then sequenced it. These are, let's suppose, all the sequences you got back. And because we're lucky enough in this presentation, let's suppose you know each sequence comes from which organism, and they're all colored by the organism that they would hypothetically come from. If we look at this little image, this would represent what our microbial community looked like. Each different color represents a different bacteria. The size of the dot would represent the relative abundance of each one of those micro, and each different circle is a different sequence, DNA sequence. So let's suppose I want to form clusters at a 97% similarity threshold. Well, what might happen in this case is that we would have only 2 bacteria. It could happen that bacteria red, which is B, and bacteria D, which is green, would be clustered together because they share a lot of similarity in their sequences in that specific region of interest. And what might happen also is that your bacteria A and bacteria C are clustered together. You might see how that can be problematic because you're losing diversity in this sample. And the reason why in this cluster it is red instead of green is because, as you can see, the red dots are larger, meaning that their relative abundance is larger than the green one, which will suggest that this red bacteria will be the dominating sequence of that cluster, and it will be what we call the representative sequence of that cluster. So this would be bacteria B and bacteria A. But you can see how that can be problematic, specifically if you're interested in fine scale diversity in your microbial community. Let's switch that up and put a 99% similarity threshold. What we see that's happening is way better, right? In that hypothetical case, I'm able to identify my 4 bacteria and I'm forming, it seems that all the good dots are going in the good circles. The only slight issue you might see in there is that I have this yellow dot that's being clustered with bacteria C. And this problem, it might not be a big deal, but maybe that in your community, it might make a big difference. So we see that it's still not an ideal situation because it's not because you increase your similarity threshold that you're going to recover all the diversity, because there are some organisms that share a lot of similarities, specifically in your region of interest, that could just not be able to be differentiated by this method. So because this isn't perfect, we shifted more recently into more complex algorithms that instead of forming these clusters of sequences that share sequence similarity, we're forming Amplicon sequence variants, or you can call them ASVs, and that's what we'll do in the tutorials today. An Amplicon sequence variant, instead of, if you want to visualize it a little bit, instead of forming this perfect circle around your DNA sequences, it's trying to form a line and really trying to grab the diversity that belongs to one organism. So we can see here that, for example, this red bacteria grabbed everything. The green one rejected one sequence, and same goes for bacteria A. So what we see here is that because Amplicon sequence variants are not just clustering sequences based on shared similarity, it also is trying to detect errors. We talked about errors before. We know they occur. We know there are problems. So one thing that ASV algorithms are trying to do is to identify an error and remove it from your data set. So that's a good thing. That's better because you're able to identify everything and remove potential errors. So today we'll what we'll use in the tutorial is Data2. It's one of the many algorithms that exist that form ASVs. Another very popular one is called Deepler, but because we'll use Data2, I'll just go over quickly how the clustering works. Since it doesn't use sequence similarity, what does it do? So to form an ASV with Data2, let me just move this. To form an ASV with Data2, there's a sort of a tree of decision that you can follow that could kind of guide you a little bit in understanding better how this clustering works. So first of all, think of data 2 as taking a pair of sequences and asking these questions. So the first question data 2 will ask is, do these sequences share at least a 90% similarity? So if they don't, we don't cluster them. If they do, we'll ask another question. The next question we ask is, so data 2 has a probability error model, and it will use that model to determine if one of these sequences is in error of another one. So depending on the threshold that's set, there's a default threshold for that, but you can play with that depending on how much you trust your data. But if the model decides that the probability that one is in error of the other is very high, we'll go to the next question. But if that probability that one sequence is in error of the other is very small, then we won't cluster them. And some parameters that are involved in this probability model is the quality score of each single nucleotide. And that's something new, right? Because we know that OTUs were only using sequence similarity, whereas in here, we're integrating the quality score of each nucleotide. So let's suppose that the probability that one is in error of another one is, the probability is very small. We'll ask the question, is it too abundant to be in error? So now we have these two sequences. We think that maybe one is just an error of the other, but then we'll ask the question, is this one too abundant to be in error? So we'll just look at the relative abundance of that sequence. And depending on the answer, if the answer is yes, it's too abundant, we won't cluster them and there'll be still two separate sequences. But if it is too abundant, we'll just cluster them together. So that's how data 2 works, but it does that for all of your sequences. And you can see how it can be sort of a long step in your analysis. It can take hours. So, and later today, we won't get to run it. I have the results already ready for us, but at least we know how it works. So just as a review, if you start with your Amplicon reads, where each dot is a different DNA sequence, and you want to form OTUs, your OTUs are only based on the similarity that is shared between the two DNA sequences, and a couple of programs that can do that for you is Research, or you can use Chime1, and then you can play with the percentage of identity. Or if you want to form, I'm so sorry, I don't know how to remove this bar. Oh, or you can form ASVs, which can be based on the quality of your nucleotides, which is what data 2 does, or it can be based on the distance between two reads, which is what Diebler uses. And also ASVs try to squeeze out the good reads and kind of get rid of the potential errors, and some programs that exist are data 2 and Diebler. Okay, so I've just talked about OTU, data 2, Diebler, but don't forget, in bioinformatics there are always a million of programs that exist that you could use. So there's a lot of other ones that exist, but they're not as popular as data 2 and Diebler, which is why I focused on them today. So you might wonder, how can I safely cluster my data? You just talked to me about all these methods that all have problems. Well, you can try different methods and try to see how that impacts your results. You can also try to better understand each method. What are each of these methods doing? And compare your results, and also the most important part, just be able to defend why you used one method over another one. So regardless of the method that you use, you'll always end up with something called a feature table. Your feature table is the basis of all of the rest of your analysis. Basically, it has as rows all of the single microbes, and each column is a sample. And inside of that table, what you get is the relative abundances of each one of those microbes. And by microbes here, I mean OTU or ASV. And so you'll see that, for example, microbe 1 is present 5 times in sample 1, for example. So this is what you'll use for the rest of your analysis. But because ASV and OTUs have these names, they're unique identifiers that are given by the program you use. These don't really talk to us, right? It's hard to make biological interpretations of just these random numbers. So one thing that you might be interested in doing is assigning taxonomy to each of your OTUs and ASVs. So now we'll do taxonomic classification quickly. So for taxonomic classification, you can use different programs that basically will try to give a biological relevant name to your OTU. You can use BLAST, which basically does local alignment of your sequences. It cuts your sequences, uses k-mers as 2-3 nucleotides, and then try to make alignments with known database. So we have known taxonomic groups, and then their DNA sequences. And when you use BLAST, it basically takes the sequence you give to it and try to see what could be a good match. But you can also use, which is what we'll use today in our tutorial, a supervised learning algorithm, such as a naive-based classifier. Basically, how it works, just quickly, a supervised learning algorithm is basically an algorithm that you train. You give it information to train the model, and then you give the model unknown data, and then it will try to classify your data based on what is known. And in this case, what is known is our databases, such as Silva, Green Genes, what Rob was talking about before, where we have taxonomic names, they're matching DNA sequences. And then what basically the classifier will try to do is calculate the probability that the unknown sequence belongs to a group, given that we know that this group has this sequence. You can also train your own or use a pre-trained classifier. Today we'll use pre-trained classifiers from QIIME 2, but ideally you use a classifier that has been trained on your specific primer region. That would give you, that would increase the accuracy of your results. If you don't have access to that, there are classifiers available for the full length of the gene. So for example, if you're using 16S, there are classifiers that are available for that. And if you're using some other genes, some other protein coding genes, depending on your research question, you might also be able to find classifiers online or classify your own, make your own classifier. And finally, just quickly, one small note about weighted classifiers. Weighted classifiers are slightly different. So in a naive-based classifier, we assume that all the organisms have an equal chance of being recovered in your final community, but we know that that's not the reality. So in 2019, this group, Kaler et al., published in Nature Communications a paper where they talk about weighted classifiers. And quite frankly, I don't know how much this is used, but I think it's a very interesting idea. And basically what they showed is that if you train your classifier based on prior knowledge about the relative abundances of different groups, you'll get a better taxonomic classification overall than if you use just a naive-based classifier that assumes that all the organisms have the same chance of being recovered in your final community. So later today, we'll just use a naive-based classifier on the full gene, but that's it for my section. So I believe I will pass the mic back to Rob, if Rob is here. Do we have any questions while we wait for Rob? Yeah.
So what's the similarity threshold? I assume it's high.
So as you saw earlier, it's not just based on the sequence similarity. So Dalhousie, the first threshold they use is 90%, and they go through that error model and then all these other questions. But and I don't think you're able to change that 90% first cutoff threshold of similarity. DeepLearn, for example, it uses distance between two reads, and the distance is 11 nucleotides, and then it's the same thing. It asks a bunch of questions depending on abundance and distance mainly, and then they're able to determine if a read is an error or not. And other algorithms, I'm not sure. But I know that for these ones, it's not just the sequence similarity. It's kind of the first question they ask. They must share a little bit of sequence similarity, and then we go through our probability model of answering whether or not these two sequences come from the same organism or not. Yes.
Are the databases like Silva updated with the latest taxonomic name changes?
Silva is updated quite regularly, just like Rob was saying earlier. I don't know when the last update was, but Silva is updated very regularly, and every time you use a classifier, you're able to see when was the last update. And also, if you use specific primers, usually you should have access to probably a classifier that has been updated lately with the correct or the most up-to-date data. Does that make sense?
Yeah.
I actually I pinged someone else in the lab because Mike Hall actually is somebody who should have been acknowledged in the slides because he helped out with some of the material too, as well as the tutorial. And he recently did an analysis for an experimental gingivitis paper. And part of that, he used Silva. Part of that involved updating the taxonomy. And well, okay, well, I'm not going to share the visual, but I asked him in our Discord, and he said, or I asked if the new taxonomy was applied by Silva. And he said, yes and no, mostly yes, often no, because he had to manually edit some of the names to make it consistent. So like he said that, and I kind of mentioned this already, Campylobacter, the now formally described, if you agree with Garrity and Oren, the formally described name has a Y, but the one that he found had an I. So the answer, I guess, is, to echo Mike, mostly yes, sometimes no. It's tough to keep an eye on, for sure.
I will let you continue, Rob.
I will continue. Thank you, Diana. That was great. Okay, so we are already at the hour. So I think I'll continue with the diversity analysis, but I will aim to make the section after lunch really, really short so that we have a balance that very much favors the actual tutorial, because that's what you're here to do. You're here to actually do hands-on learning. So I'll try to make this a little bit more concise. Okay, so microbial community analysis. Well, what's a community? Informally, you might think of it as the group of things, species, whatever, that occupy the same location at the same time. There's an extension of this notion, which is the meta-community, which is the group of things that occupy the same region at the same time. And the reason people think in terms of meta-communities is because even if you don't see something in a given place at a given time, it doesn't mean that it can't be there. Just maybe you didn't observe it there at that time. And if you look more broadly in the region, whatever the region means, maybe it's the mouth, maybe it's a forest, then you would find it there, right? And so people have different ways of describing and modeling the processes that influence meta-communities. Notice that community interactions are hypotheses. So you say, oh, I found a bunch of stuff here at this site at this time. Doesn't mean they're all talking to each other, interacting, trying to help each other, trying to kill each other, whatever. So it does not necessarily mean that they are meaningfully interacting with each other. Okay, so two fundamental concepts, richness and diversity. They're related to each other, but there are some differences. So richness in its simplest form is the count of things. And we'll get to what things are in a moment, but just, you can say species for now. We'll touch on this in more detail later. So your formula for richness is simply the number of species you see. Diversity takes this notion and augments it with some notion of evenness, right? So Shannon diversity is the classic example of this. And what you have, there's that R again, right? Because that's each of your species in turn. And in each case, your diversity is going to depend not only on the number of things, but their relative proportions. And that's what PI means. So P sub I is the proportion of species I, right? So, okay, Cetobacterium is 20% of my entire sample. So P sub I for Cetobacterium is 0.2, right? So diversity will take those relative measures into account. Okay, so richness and diversity of what? I said species. Doesn't have to be species, but it could be species. It could be genera, could be phyla, could be OTUs if you want to use OTUs, don't. It could be ASVs, could be unique sequences, could be functional genes, could be a whole bunch of different things, right? Depending on how you want to describe your sets, at what taxonomic level, at what functional level. Not all approaches make sense for all types of things. So there are a lot of diversity measures. My PhD student, Donovan Parks, a few years ago, did a comparison of the performance of 40 different beta diversity measures. So there's a lot. You don't need to use all of them, but what's really important, what's most important, is to understand the fundamental differences in the ways they operate. And so one important difference is that some methods work with observed data and some work with estimates. The reasoning here is that if you're taking a finite sample of a population of a community, then you're probably not going to observe everything. Maybe you're happy with making your calculations and your inferences based only on what you see. But maybe if you're trying to understand diversity, you also want to say, okay, well, I've got this level of observation. This would imply that I have even more stuff that I didn't see. So let's try to model that. Non-phylogenetic versus phylogenetic, very important. So if you define things like species, a basic measure like richness or the Shannon diversity does not care how closely related those things are, if there is a way to define their relatedness. So a non-phylogenetic method counts things. A phylogenetic method will consider the relative similarity. So for example, let's say you've got one community which has 2 different strains of Salmonella, and another community that has a strain of Salmonella and then some weird exotic archaeon. Which is more diverse? Well, if you're using a non-phylogenetic method, then if you're defining your things in a certain way, they're equally diverse. But a phylogenetic method will say, whoa, 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 no, no. Look at the branch lengths, right? The Salmonella are really, 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 really similar to each other, whereas there's this obnoxiously long branch length connecting the archaeon with Salmonella. Very important distinction. The other really crucial distinction is the one that I already alluded to, which is unweighted versus weighted by abundance, right? So richness, unweighted. Shannon, weighted by abundance. So 2 main types of diversity that are considered in microbiome studies, not the only 2 by any means, but in terms of microbial ecology, these are the ones that you would typically expect to see. Alpha diversity is when you look at the diversity of a single site, right? So I've collected this sample from the subgingival plaque in my mouth. I would like to get some notion of the diversity at that site. And so I've got a little matrix there, a little grid, and I'm showing you examples of richness versus diversity measures, right? So that's the, just number of things versus considering relative abundances. And then down the side, we've got non-phylogenetic versus phylogenetic. So the top row, the non-phylogenetic, is exactly what I showed you a couple of slides ago, species count versus Shannon. On the bottom, we can incorporate the phylogenetic dimension and let's say build a phylogenetic tree, as with the Salmonella and Archaea example I just mentioned. So for richness, you can do unweighted phylogenetic, scare quotes, diversity, because you're not considering the relative abundances of things, you're just counting them up, weighting by the phylogenetic tree. With abundance weighted phylogenetic diversity, now you're considering the phylogenetic tree and you're considering the relative abundances of the different things you're looking at. So in the first instance, it doesn't really matter whether it's 50-50 Salmonella. In the second instance, it matters whether it's 50-50 or 70-30. Okay, so this is a useful framework to think about, and not to anticipate the future too much, but if you're concerned about trying to assess the diversity and worried about what different measures might give you, then this is a good sort of mental framework to say, well, I want measures that are likely to give me different answers, right, in principle, to see whether they agree. So this is a useful way to think about that. Okay, and this is just an illustration of what I already mentioned in terms of phylogenetic diversity. All right, so what can we do with alpha diversity? Well, we can relate it to things. So under certain conditions, under certain criteria, or certain types of communities tend to be more diverse than others. Well, what does that mean? And so on the left, I've got an example from a soil microbiome paper from quite a few years ago, but it's a nice illustration nonetheless. And we've got two figure panels there. Notice that the x-axis in both cases is soil pH. So the authors were trying to understand the relationship between soil pH and some notion of diversity, if one actually exists. On the left, we've got a phylogenetically weighted method. And you can see that, and this is consistent with the hypothesis where neutral pH roughly is expected to have highest diversity. And according to the model fit, yes, highest around 7, maybe 6.5, drops off in either direction. On the right, we see the same analysis, but instead of using a phylogenetic tree, it's the number of phylotypes, which is basically a surrogate for species or genera or something like that. So you can see that although the strength of the relationship differs, the relationship overall remains the same. On the right, we have a similar idea, but now we're looking at, rather than quantitative predictors, we're looking at categorical predictors, right? So in this case, we've got looking at the diversity of microbial communities in different types of newborns, right? So we have very low birth weight versus control, and very low birth weight is indeed very low. Trying to understand the dissimilarities in community composition. And so you can see that the Shannon diversity is summarized with those box plots. Just two out of a multitude of examples. Okay, so that's alpha diversity. The other one that you'll typically see used is beta diversity. And this is where you take two samples, two sites, and you'll compare them against one another to get some sort of measure of similarity or dissimilarity. So we're talking about diversity, then more dissimilar communities will have greater beta diversity. In general, you would expect identical communities to have a beta diversity of 0. If you compare something with itself, you expect it to have no differences, obviously. That framework that I outlined before, phylogenetic, non-phylogenetic, weighted versus unweighted, applies here as well. And there's, in many cases, direct analogs between alpha diversity measures and beta diversity measures. So a couple of examples. The Jacquard is unweighted, non-phylogenetic. I'm not going to walk you through the formulas in detail. The basic idea is you have this stuff, I have this stuff if we're two communities. Here's the stuff we share in common relative to the total set of stuff we have, right? So if we overlap in our species composition, then we will tend to be very similar. And in general, if you want to change a similarity score into a diversity score, you subtract the similarity score from 1, right? So this is similarity, but we can change it into a diversity or a difference measure. Okay, Bray-Curtis, also non-phylogenetic, so you still need to define things, but now it's weighted by species abundance. Okay, and so again, not going through the formula in detail, but the quantity C sub ij is count based. Okay, and so is S, right? So now you're looking at counts rather than just presence absence as you are with the Jacquard. Unifrac, which many of you may have seen already, some of you may have used, is a phylogenetic extension of the notion of diversity. And again, by no means is it the only one, by no stretch of the imagination, but it follows this general principle that we want to consider, for all of the things we're looking at, the degree of overlap. And so there's a couple of contrasting examples there in front of you. Each of the communities is indicated with a different symbol. So one community is red squares, the other community is green circles, and the phylogenetic tree relates all of the different things that were found in each of those communities. On the left, the phylogenetic tree intermingles the red and the green. And so maybe the clade on the left is Salmonella, right? And so each of the communities has something in Salmonella. On the right, the communities are completely phylogenetically separate, right? So you can see that one clade in the tree has nothing but green circle, and the other clade in the tree has nothing but red. The branches are labeled or colored depending on whether they cover one community only or both. And so, purple corresponds to branches that cover only one community. So on the left, you see that you've got purple branches that lead to the red squares and purple branches that lead to the green circles. But notice that the parent branches are black. Black branches are branches that cover members of both communities. So notice, let's take a look at the leftmost black branch in that leftmost tree. It's black because if you go down, if you look at descendants of that branch, you see both communities represented. On the right-hand side, there is no branch in the tree apart from the one that leads to the root. There's no other branch in the tree that covers both communities. Unifrac says, what is the unique fraction of branches? Okay, so the Unifrac measure informally is the purple branches, the total length of the purple branches on top, divided by the sum of the purple and black on the bottom. So purple and black is the total size of the tree, the total branch length. Purple is unique. If all of the branches in the tree are unique to one community or the other, as they are on the right, then Unifrac distance is maximized, it's equal to 1. Oh, and sorry, I forgot to mention that there are weighted and unweighted versions of that, depending on whether you're looking at just presence absence or proportions. You can build matrices that show the beta diversity across many, many different samples. And so this is just a simple example from this cute little animalcules app or application that can do all sorts of little visualizations and analyses of microbial community data. And so rows and columns are different samples, and then each matrix entry indicates the diversity, the beta diversity of that pair of samples. So you can see the diagonal is white because each sample is identical to itself. But look at the off diagonals. The darker purple indicates the most dissimilar communities in the set. So this starts to give you a sense of which communities are relatively similar and relatively different from each other. Once you have a matrix, the world is your oyster. So you can do all sorts of clusterings. And I'm not going to go in these in detail, but the idea is you take your matrix and you can do some form of dimensionality reduction, some form of ordination analysis to get your matrix, your samples in a plot. That allows you to visually compare your sets of samples and see whether there's any sort of clustering by body site or by soil type, for example. You can also do hierarchical clustering to look at the relative degrees of similarity. This is a nice intuitive example because you can see the colors representing the taxonomic breakdown of each of the corresponding samples, right? We've got our different phyla listed there. The tree on the left is not a phylogenetic tree. It's a cluster tree showing the relative similarity of each of our samples. So notice at the top there, HC2 and HC1 have very, very similar taxonomic profiles, and consequently, they are clustered together in this hierarchy. All right. So many diversity measures, a ridiculous number of diversity measures. Which ones should we choose? Well, it comes back to that point I made before. If you're going to choose a couple, don't choose 2 that are expected to give very, very similar results. Choose 2 or 3 that are built on different foundations, phylogenetic versus non-phylogenetic, weighted versus unweighted, and then see if the results are consistent. This is a paper that Donovan did back in 2013, where he looked at about 40 different beta diversity measures. And so this is actually how similar their predictions are. You can see that Bray-Curtis at the top is very similar to many others that are following the same paradigm. So normalized weighted unifrac, Kolzinski, Sorgel, and so on. Each of these is published separately, but you can see that they tend to give very similar results. So don't take 2 of those, compare them and say, wow, they give the same answer, because that's not really surprising. If you want to do real comparisons, take your Bray-Curtis or your weighted unifrac and compare against some that are in other blocks in this clustering.